All right. It is the final Saturday. Let me just admit the people who are joining. Yeah. For a couple of minutes, let me just briefly explain what we are going to do today. And during that time, I'll admit whoever is trying to join the meeting. This also brings us to the final week of our interaction with respect to the functional genomics course. So today is the final interaction session. So for today's session, what I have done is, as we have been doing in our set format, we will solve a previous year's question for which were basically for the week for content in last year. So we are going to basically solve them. After that, what I have done is, on the request of some of the learners who are continually attending these classes, I have dedicated part of this lecture to discussing newer genomic editing techniques, right? So they are basically techniques wherein CRISPR has somehow been modified to like further edit the genome in a more efficient and maybe less off target site interaction specific manner, right? Very briefly, I have kept three major techniques, which are base editing, prime editing, and the final one, which was okay. Base editing, prime editing, and which was the final one? It was paste. Okay. I that particular technique. So yeah, so these three techniques we will touch upon. We will understand what is the principle behind these techniques, how they are better than the previously ongoing functional genomics or genome editing techniques. Also, if we have time in the end, I will discuss one or two new diagnostic techniques using these tools that have actually come up. Okay, so not wasting much time, let us begin with today's lecture. If anybody would be joining, I'll be happy to let them enter the meeting, or if you guys get a chance or get this option, you might admit your co-learners, right? Let me first begin with the previous year's assignment for week 4. Yeah, so we again have 10 questions. We will go through each question. And okay, sorry, I have not shared my screen. Sorry for that. Yeah, the recording is on and I need to share my screen. Yeah. All right. So we are beginning with the previous year assignment for week 4 content, and like always we have 10 questions. We have options for those respective questions, and we'll keep on solving those questions. We'll read all the options. I'll give you a moment to maybe judge or decide which one could be the right option. We'll discuss which one the right option is, and if at all there is any concept linked to it that was not very well, or it was not stressed upon or in the original video. So for that, we will take a little time. We will explore those concepts and then move forward. Right. So for week 4, the first question is, which among the following options will not be much useful to study comparative genomics? So the essence of week 4 was comparative genomics, at least the lecture part, right? So which one is not useful to basically study this area? So is it whole genome analysis, or gene clustering by function, or antibody screening, or gene sequence variation studies? So which one do you think won't help you to study comparative genomics? So let us move on to the answer. It is actually antibody screening. Why? Because all these other things, for example, if there was a GWAS or whole genome analysis study, what will it give? It will basically give you an output such as a whole genome sequence of whatever individual or organism you were targeting, right? If you perform gene clustering by function, what does it mean? That, for example, you have multiple organisms or multiple individuals. Let us go with organisms, because over here we will deal with different species, right? So, for example, you have different species, and within those species, you know that everybody has a glucose uptake mechanism. So you want to understand what is the gene responsible for glucose uptake mechanism. So the function you have dedicated, that it is basically glucose intake mechanism, is the kind of function you want to study, right? But can we cluster genes according to that? Are those genes conserved across all species? Were the primitive or the invertebrate animals, did they have a different kind of uptake mechanism than, say, the vertebrate animals? Or do mammals have different kind of uptake mechanism than maybe apes? Right. So such things can also help you study comparative genomics. The final one is gene sequence variation studies. So gene sequence variation studies is something that we had loosely talked about when we were talking about MSA or multiple sequence alignment as well. So in my last class, when I discussed multiple sequence alignment, we basically talked about the homology or complementarity between sequences, right? So if the same bases are retained across many such sequences, it basically means that they are homologous, or they have similar sequences, right? These similar sequences could basically indicate that maybe these two organisms have similar function or similar origin, maybe that based on that phylogenetic trees are built, right? So gene sequence variation studies are also helpful in comparative genomics, whereas antibody screening is something that is done in a immunological or molecular biology perspective, but it will not really help you to understand comparative genomics, right? So, comparative genomics is a field of biological research in which researchers use a variety of tools to compare the complete genome sequences of different species. By carefully comparing characteristics that define various organisms, researchers can pinpoint regions of similarity and differences. Also, please excuse me if anybody would have said anything up till now. I just realized that my speaker was off. So please excuse me if I did not listen to what you were saying. I have turned it on. I'll be able to use it now. Right? It's completely skipped my mind. Okay. So anyway, I hope it was visible throughout, right? Anyway, I'll be moving forward to my next question. Okay. So before the next question, actually from the resource wherein I have picked that definition, that is National Human Genome Research Institute, I had also basically picked in this particular fact sheet, which basically explained how comparative genomics is a good approach to study a few areas or fields, right? So over here, they have basically said that common features of different organisms, such as humans and fish, are often encoded with the DNA evolutionarily conserved between them, right? So comparative genomics basically helps us understand conserved regions of the DNA. How will these conserved regions help us? Or how will this understanding help us? Because it will tell us that maybe if there was this feature might have been selected during evolution, right? Maybe if some organisms might not have expressed this particular DNA. Just a moment, let me admit people. Yeah. So if maybe a particular organism would not have been expressing this particular DNA, they might have been negatively selected, right? Or they might have been wiped out. Then looking at close related species, such as humans, chimpanzees, show genomic elements that are unique to each other. Despite similarities between the genome, there are also some dissimilarities. How will understanding these dissimilarities basically help us? Because they will actually explain to us why maybe apes who are our direct ancestors, right? Just before us, we have basically evolved from apes. That is the consensus, right? So, how we are so evolved that we have literally flourished. We are kind of owning the planet, right? While apes, they are left so backward, right? So these differences between genome also helps us understand what basically favored us, what in our genome favored us over the other species, so that we could flourish this much, right? Then genetic differences within one species, such as our own, can reveal variants with a role in disease. So this is kind of what we do when we do genetic analysis or positional cloning, right? So we find that in a normal individual, this is what happened. This is the sequence, but in case of a diseased individual, this sequence looks different, right? So that is how we identify different diseases or disease-causing genes, right? Okay. So that brings us to our second question, which is the term comparative genomics refers to a field of study in which the genomic features of a different organism are compared. Which one of the following options may not be a direct outcome of comparative genomics? So it is again something that is related to comparative genomics. Just a moment. Yeah. So I'm back to my presentation. I just read out these questions, and the options for the question of which one might not be a direct outcome of comparative genomics is identification of regulatory elements of the genome, or understanding evolutionary changes in the genome, or phylogenetic positions of species, or epigenetic changes. So we just discussed what comparative genomics are. What are the kind of things that help us basically study comparative genomics? And if we actually do study comparative genomics, what are the kind of outcomes that we get? Right? We understood all these prior to this question. So, which one do you think would not be a direct outcome of comparative genomics? Okay. Yeah. Okay. So, jumping on to the answer of this question, okay, my slide is on. Yeah. So it is epigenetic changes, right? For example, we have just discussed what comparative genomics is, right? So we know that comparative genomics, if we basically compare the genome sequences of two individuals or organisms or different species, we can basically identify regulatory elements of the genome, right? We can identify the evolutionary changes in the genome. We can also list species in the phylogenetic trees, right? So, based on how much sequence similarity there is, maybe between an ape and a bird and an ape and a reptile, they would be placed at different positions in a phylogenetic tree, right? But can we look at epigenetic changes just by comparative genomics? So, no, right? So epigenetic changes involves not just looking at the sequence of a person's genome or DNA sequence, but it also involves other environmental factors or chemical modifications that go on within this particular DNA, right? So the sequence might still be the same, but due to some environmental factors or some chemical moieties that might be coming and influencing this particular sequence of DNA, there might be some modifications that we cannot monitor using comparative genomics, right? So epigenetics, let me explain what it is also sometimes known as epigenomics. It is basically a field focused on changes in DNA that do not involve alterations to an underlying sequence, right? Uh, so somebody is trying to join the class. Let me. Yeah. Okay. So, like I just said, epigenetics is a field that is focused on changes in the DNA that do not involve alterations in the underlying sequence, right? The sequence might be exactly the same, but the DNA letters or nucleotides, as we know, and the proteins that interact with this DNA, proteins such as transcription factors, maybe, or other polymerases that are being recruited to it, we can have chemical modifications that changes the degree to which the genes are turned on and off, right? For example, if we talk about proteins such as histone, if the DNA is tightly wrapped around a histone,
Yeah, just
it
see the screen. I'm not able to see the screen. I don't know about other two.
okay.
I tried to join and rejoin. I'm not able to see the screen. Okay.
Is that the problem with everybody? Okay. Wait, let me just stop sharing and I'll share again. Maybe that resolves the issue. Is it okay now, Ajay?
Yes, madam. Now,
Okay. Okay.
I don't know why others are not complaining.
Okay. So right now it is visible to you, right? Okay, I am hoping it is. Uh, Maybe just give me a moment. And I will try to sign in with a different device so that I know when this problem is coming up. Okay. Yeah. Okay. So hopefully I'll know when this problem is coming up now. I will stop sharing and share again. Yeah.
Now visible. It's
Okay. So what we were doing was we were just discussing this question wherein they had asked which one of these given options would not have been a direct outcome of comparative genomics, right? And we narrowed down the answer to epigenetic changes. And then we were basically discussing what epigenetics in general is and how these changes will basically not affect or not be able to be visualized using comparative genomics technique, right? So over here, so I was giving an example about a protein which is histone. So as you all are already aware, what histones do is that histones help wrap this enormous length of DNA into a very condensed form, right? So that when it is not replicating, it is in a condensed state, and it is basically useful to pack that enormous amount of DNA into a smaller compartment, right? But if this histone wraps DNA in such a way that it is not at all available to the replicating or transcription machinery to basically transcribe an mRNA or replicate, it will basically pose uh, a different kind of scenario, right? Wherein this DNA is present, it is not mutated. The sequence is very similar to what it would have been in case of another individual or another organism. But since there is some defect, some epigenetic change that is not allowing this DNA to unfold from this particular histone, there will be some other kind of defects, which one might not be able to assess using comparative genomics or just the gene sequence, right? So that is one example. Okay, moving on to question number 3, which says, which one of the following options is a true statement for homologous recombination process? So a similar question we dealt with in the last class also. The options for this one are, it is a type of genetic recombination in which nucleotide sequences are exchanged between two similar or identical molecules of DNA, or it occurs during the formation of the egg and the sperm cells, or it mostly occurs between homologous chromosomes bearing distinct markers surrounding the exchange region, or all of these.
All
So,
of these.
yeah, so yes, we discussed a very similar question in the previous class also, and the answer to this is all of these. So, let us briefly revise what homologous recombination is. So, in homologous recombination, what happens is, first of all, this process takes place during meiosis, right? During meiosis, what happens is that gametes have to be formed. During this formation of gametes, what happens is that in our own genome or in our own DNA, we have two copies of genes, right? One copy is from our father and one copy is from our mother. So the paternal copy over here is basically listed as or drawn as green, and the maternal is drawn as mauve or purple, right? So what happens is that each of this chromosome is basically alike. So for example, we are talking about chromosome number 1. So both these are chromosome number 1 only, right? But this particular copy of chromosome has come from our father. This particular chromosome has come from our mother. What happens during meiosis is that this chromosome has to be reduced into half. So right now there are two sister chromatids that are present in each chromosome, right? So this green, both the arms are sister chromatids among one another, and purple, both arms are sister chromatids among one another. These sister chromatids have to be reduced into one chromatid per individual, right? So what happens during homologous recombination is that before this reduction happens, what happens is that these non-sister chromatids, that is one of the father's chromatids and one of the mother's chromatids, they come together and they basically form a junction across one another, through which basically the recombination occurs, right? So they will physically be joined at one locus, and ahead of this locus, they will exchange their DNA, right? Why is it important? Because it basically helps bring in heterogeneity into our genes, right? Or it brings in new features in our gene. So we are not the exact replicas of our parent, right? We are kind of somewhat like our father, somewhat like our mother, right? We are not the exact replicas. It is because of homologous recombination. So this is what the recombined chromosomes look like. Also, like I discussed in the previous session, where does homologous recombination take place? So it basically takes place in meiosis 1. So in this meiosis 1, there is a stage that is basically known as prophase 1. In prophase 1, there are different stages, some in which the chromosomes will come together. The sister chromatids will basically form this conjunction or this joint formation. And then during the Pachytene stage, which is in prophase 1 of meiosis 1, this crossing over actually takes place, right? After meiosis 1, what happens is that the resulting cells, the resultant is that we have two nuclei, one of which has a recombined set of chromosomes, and the other also has another recombined set of chromosomes, right? After this, meiosis 2 happens, which is basically the reductional division, wherein these chromosome numbers will be reduced to half. During this reductional division, after this reductional division, we have 4 daughter nuclei, each of which have just two two chromatids, right? Not sister chromatids, just two chromatids that are present. Okay. Anyway, coming back to multiple sequence alignment, wherein we spent a lot of time in the previous session. So a multiple sequence alignment analysis helps us to understand which one of the following: ancestral relationships, regions under lesser selection pressure, regulatory elements in the genome, or all of these. So this is also a fairly simple question. Yeah.
Again, all of these.
Answer. Yeah, all of these is actually the answer. So as we talked, multiple sequence alignment is just one tool which will help align similar sequences, right? Or align just any sequences. Whatever regions are homologous to one another will align. So this alignment will help us to extrapolate data about ancestral relationships, right? So for example, phylogenetic trees are built upon the data that is produced from multiple sequence alignments. There are other things such as we can identify regions under lesser selection pressure. What does this mean is that these regions, how will they be? Will they be conserved or will they not be conserved? The regions which are under lesser selection pressure. So probably these regions will not be conserved, right? So we will understand that, okay, it is all right, even if this particular region has A or T or G or C, it doesn't really matter, right? Because in different organisms, even closely related organisms maybe, they are varying. So maybe this particular consensus in this particular region, consensus of the sequence is not required for an organism to be positively selected, right? Then regulatory elements in the genome can also be identified. So for this, I don't think I have put in that illustration, but if you remember from Sir's lecture, Sir had basically explained, I think in his final lecture, how the density of the genes have decreased, right, over this evolutionary period. So what I mean by density of the gene decreasing is basically that, okay, I'm unable to annotate. Yeah. For example, in a yeast, if I say if this much length of a gene we are of DNA we are talking about, this much DNA will have coding regions only, right? So this entire sequence will have coding regions, right? If we talk about after yeast, if we talk about Aves, maybe the Aves will have coding regions in most of the DNA segments, not maybe all of them, right? If we talk about just maybe 1 kb of a fragment. While if we talk about humans, so please forgive those peaks because that is not intentional. It is just something my mouse is doing. So in humans, maybe only this much part of that 1 kb fragment is coding, right, on which I have drawn those triangles or those rectangles. So from this, we basically understand that although all these fragments are 1 kb in size, right? In case of a yeast versus an apes versus a human, the gene density is much more in a yeast than a human. That is why the humans have a larger genome size, right? So this genome size is because of presence of more regulatory elements between them, maybe, right? So there is more non-coding region, a lot of which will also be regulatory elements, right? So we will basically also be able to identify regulatory elements in the genome of individuals. So the answer naturally is all of these. So the output that is generated by multiple sequence alignment can be used to infer homology and evolutionary relationships between the aligned sequences. Therefore, all of these options will be correct. Okay. For the fifth one, they ask the field of metagenomics involves which one of the following. So this is a little tricky. Please go through the options. The study of genetic material recovered directly from environmental samples, computational analysis of the human genome, sequencing of genome of the gut microbiota, or sequence analysis of transposable elements of the human genome. So which one among these do you think is the correct statement for metagenomics? Okay. So coming on to the answer, A, right. So that is actually correct. I thought people might be confused between A and C, right? Because that is a very genuine doubt that a lot of people have. Anyway, you got it correct. So the answer is in fact A, which is the study of genetic material recovered directly from the environmental samples, right? So when we talk about metagenomics, we talk about the genomics of the entire environment of an individual, right? For example, if we talk about human metagenomics, we will take samples from the human, from the gut of the gut microbiota sample of the human, and from the environment in which that human being is living, right? So it will encompass the entire range of these samples. So metagenomics is also referred to as ecogenomics. Why? Because environmental has to do with ecology. So from that you can remember ecogenomics, in which basically involves genome analysis and DNA barcoding of environmental samples, including environmental microbes. So let us briefly take a look at how what is the workflow for a metagenomic analysis. So in metabolomics, what is done basically is that microbes or in the environment, they are basically collected, they are sequenced, right? So the metagenomic DNA is isolated, that is amplified, they are then sequenced and they are analyzed, and then they are basically also studied with reference to other databases that are already available to us, right? If we look at this particular paper by Zhang et al., what happened was that what they have done in metagenomic analysis is that they have basically taken the environmental microbiome, right, from maybe a river. And then they have grown it on a culture plate and they have basically screened for resistance. For example, they have applied multiple antibiotics on this particular plate and they have checked which kind of resistance, which kind of bacteria or microorganism is actually showing. And then they have basically sampled them and sequenced them and basically kept this or fed this into a database. So for such studies wherein maybe there is some kind of a pollution, some kind of toxic chemicals that are present in the environment, or the microbiome of a water body is such that it actually causes a lot of people to be susceptible to stomach infections, right? So this kind of a screening can be done over there. Also, there are other uses of metagenomics. So this particular slide I have picked up from Mammoth Biosciences, which is one of the companies that is also enabling researchers to perform metagenomics. So they have basically identified some basic utilizations of this metagenomics technique, right? So, for example, what metagenomics or how metagenomics can help us in research, both basic research and translational research, is basically that if we basically identify the genome of these multiple bacteria, microbes, etc., we will be able to get new CRISPR proteins, right? This will help us to expand the CRISPR toolkit and improve genome editing. Also, there might be some biosynthesis pathways that we are missing right now, right? If we actually could study the genome of these organisms, so we could, their genome could help us understand how basically maybe some resistance is being developed by a certain bacteria, and that might lead us to new biosynthesis pathways, right? Also, new kinds of viruses, antibiotics, etc., could basically be acknowledged or it could be discovered using such techniques. So, based this slide, I would want you to remember when we talk about newer genome editing techniques, that is the second part of my talk today. So in those, there are multiple new maybe transposons and integrases that have been discovered using these metagenomic techniques, which are actually being used to modify this CRISPR and to better this particular technique that is already existing. And it is really helping researchers make more and more advances in science, right? So it is a very suitable technology, high throughput technology. Okay. Which brings us to question number 6, which says which one of the following statements is incorrect with regard to phylogenetic trees? So the options are: they represent facts since supported by functional genomic data. They represent hypotheses and supported by functional genomic data. The branching in a phylogenetic tree reflects how species evolved from series of common ancestors, or rotating a tree about its branch points does not change the information it carries. Which one do you think is incorrect? Okay. So the incorrect one is the first one. So an easy guess when you narrow it down to two options, right? So the two options could either be A or B. Why would that be possible? Why would that be the case? Because these two options are basically saying opposite things. So only one of them can be correct. One of them says that they represent facts, which is supported by functional genomic data, while the other says they represent hypotheses, which is supported by functional genomic data. When there are two very absolute statements, you can narrow down your answers to them, that one out of this might be correct or might be the answer, right? So phylogenetic trees include are indeed hypotheses. They are not definitive facts. The rest of the options that are mentioned over here are indeed correct. Okay. So just for instance, I have basically kept the example of one particular phylogenetic tree, which presents two hypotheses. Right? In this, there are the same 1, 2, 3, 4, 5, 6, 7 uh, 7 different species or 7 different animals have been used, which are sharks, ray finned fish, amphibians, primates, rodents and rabbits, crocodiles, and dinosaurs and birds. So these 7 have been plotted on this particular phylogenetic tree based on whether or not they are related to one another and how closely they are related to one another, right? So the hypothesis 1 basically looks like, if you see the basic arrangement of them is the same, right? The order of arrangement from sharks to dinosaurs and birds is same in the first hypothesis as well as the second hypothesis. So that is just the case in this particular tree, right? Sometimes this arrangement is also different in different hypotheses. But where the difference arises in these two hypotheses is whether the sharks and the other individuals that have a bony skeleton, whether they have basically diverged from one common ancestor, or whether the vertebrates first diverged into two common ancestors, one of which gave birth to both sharks and ray-finned fish, and the other basically gave rise to these other these other 5 individuals, right? So where the difference is that whether the
Sorry,
bony
madam.
skeleton, yeah.
Complicated, madam. Please, make it simple. It seems too much complicated.
Yeah. Okay. So how we can understand is that it is, if we
First,
look at this first hypothesis, yeah,
my English is not so good. When there is a hypothesis, means is it a just assumption?
yeah, it is
Hypothesis.
an assumption based on the sequence that we have gotten, right?
And
So it is an assumption.
how these assumptions they differ from the fact? Because when we prove
Yeah.
the assumption, it becomes fact, na?
Yeah. So how it can actually be proven is if we basically find a connecting link, maybe, right? That is one of how basically evolution has been justified,
That's why I was
right?
confused. Both the options seems correct. If
Yeah, both
somebody
of them
thinks
seem
hypothesis and
yeah.
later on it is proved, they become fact, no?
Yeah. Yeah.
So
So
this hypothesis
the
is
only
proved.
difference between the two,
One
yeah,
one of the hypothesis is proved, so it becomes fact. So at
yeah,
that time, same question was asked, suppose 10 years back, 20 years back, or 100 years afterwards.
hmm,
Same question is asked to a student 10 years, 20 years, or 100 years afterwards. So
yeah.
that time, statement will be the hypothesis instead of the fact. So why these confusing questions are given just based on the English? It is not
Yeah,
based
yeah,
on
but
that's what
yeah.
my question is. It should be based on your functional genomics, not based on the English. Because there is
Yeah,
a
so that is actually one point, but there are very minor differences between a lot of things. For example, there is a question that is pertaining to speciation. When we come to that, you will understand that just understanding the concept of speciation is also based on very, very minute differences. So when the differences are very minute, you have to say that correctly, right? Uh, that is a very good example of what you are actually saying. So once we come to that particular question, it is in today's lecture only. You will understand why basically these minute differences are important, right? So I understand that if it is not a native language, just because of grammatical error or grammatical misunderstanding, we could get the answers incorrect. But what we have to understand is that it is one of the universal languages in which research is being done, right? So, in order for us to actually understand whatever is being told to us, also in a paper, for example, science, right? Science papers are very, very concise, and they in maybe one paragraph they will say a lot of things, right? If you don't understand each line, it is very hard for you to grasp the gist of that paper. So that is one of the drawbacks also. But since we want the language to be universal, the language of research to be universal, that is one of the things we will have to let go, right? Okay. So, in the first hypothesis, what happened was that they basically said that maybe there was a vertebrate, right? From this vertebrate, there were two branches that arose. One branch basically led us to sharks, which basically have a cartilaginous skeleton. The other branch basically led us to all these other animals, which all have bony skeletons, right? So, ray finned fish, amphibians, primates, rodents, and rabbits, crocodiles, as well as dinosaurs and birds, all of them have bony skeletons, while sharks have cartilaginous skeletons. Further, this branch that was dividing or clustering this particular group of animals which had bony skeleton, it further got divided into one branch that just had the ray finned fish and another branch which all all these animals had four limbs, right? So amphibians, primates, and other such animals that all had four limbs each, right? So this is how, and then again, they were divided based on whether their eggs were amniotic or they were covered in an amniotic sac or not, whether they had hair or not, whether they had two post-orbital fenestrae. So these are unimportant for this discussion, right? So what they meant to say was that first hypothesis says that this is how the divergence happened. While in the second hypothesis, what happened was that they say, or other than this, what could have happened is that maybe there was a vertebrate individual which was a common ancestor. Out from this individual, what happened was that two branches evolved. One branch basically led to sharks and ray finned fish. The other branch led to amphibians, primates, rodents, crocodiles, and dinosaurs, right? Now, in this particular branch, wherein one was leading to these fish and the other was leading to these four-limbed animals, separately, two evolutions happened or two kind of mutations happened, wherein ray finned fish developed this bony skeleton independent of these individuals developing bony skeleton. So, what they mean to say is that the bony skeleton was obtained both in ray finned fish and these other individuals. But over here, it evolved independent of these particular individuals, right? So there was not a common ancestor who had bony skeleton. They all evolved separately, right? So some people do believe this hypothesis, and there might be some indications that this hypothesis is true because obviously sharks and fish, sharks are also fish, right? So they, if you think about it, they seem to be closer to one another than fish and amphibians, right? But again, this might also have been the case that sharks and sharks evolved separately. Then, in a separate set of evolution, ray-finned fish diverged, and some of these common ancestors only gave birth to other kind of individuals like amphibians, dinosaurs, etc. Right? So we don't know which one of these hypotheses is actually true. So what is understood, like what is taken as an understanding in biology is that which one will be considered as a correct hypothesis. So can you answer that? Which one do you think should be considered as the correct hypothesis? Like you may believe differently, right? There might be. Yeah.
Shark with the whale, and I think hypothesis one is correct. But I think in hypothesis, instead of shark, you should write down whale. Means the one who has written that shark. The shark is more
okay.
Shark is a mammal, and ray-fin is in a aqueous form. No?
Whale is actually a mammal.
Shark, they don't have differences. But whale and ray-fin have differences. Ray-fin fish and whale that have enormous difference, and that's why it comes into vertebrates. And if you exchange shark with the whale, then hypothesis one seems to be more correct.
Sharks cannot be changed with whales, right? Because again, whales are mammals.
Shark is mammal? I don't think.
No. Whales are mammals. Sharks are fish. Okay. So and
fish.
This is something that
if we replace with the whale, so hypothesis one will be correct.
No, so it cannot be exchanged with whales because whales cannot be this far away from rodents, right? We are closest to, we come under primates, right? So whales will come closer to rodents and primates than fish. Okay, so that is a whole different discussion. But this is something that is not designed by me personally. So it is basically designed by people at Berkeley, and they are experts at this. And it is actually a very simple representation. And changing sharks with whales is a very, very incorrect way to.
Sorry, madam, to interrupt. On the basis of presence of air, rodents and rabbits, but that cannot be present in the whale. So that's why my confusion was there. Ki instead of shark, there can be whale. And what people had designed might have their own view. I was just explaining my view. Okay, madam.
Even then, whales do have an amniotic egg, right? So that is something that is not there in case of fishes. So so this is actually a very simplistic diagram. Between this, so many other species also come in, right? Just for the sake of making you understand how basically it is picked and chosen, they have simplified it to the level that it was possible. And what they have said is that obviously without concrete facts, we cannot say that this hypothesis is true and this hypothesis is incorrect, right? Or it is untrue. But if we actually have to pick one, right? For example, just for the sake of teaching it to students, we cannot go on and teach like 3, 4, 5 hypotheses for every phylogenetic tree, right? So we can't say that in this hypothesis we think this is possible, in this we think this is possible, in this we think this is possible. Maybe at an advanced stage, yes, but at an early stage, when you are taught things at a very simple level, how will you pick one better hypothesis? So for that, what is a basic consensus or a basic understanding is that the principle of parsimony. So have you come across this term, principle of parsimony? So if not, what it means basically is that we will basically choose the simplest whatever is the simplest understanding or simplest outcome, right? So whatever will simply explain what happened is what we will choose. So in this case, hypothesis 1 simply says that vertebrates divided into these two branches, then bony skeleton developed, and then they were divided into these 5 kinds of branches, right? Further, after that, forelimbs developed. After that, they were divided into whatever other branches. While hypothesis 2 says that vertebrates divided into 2 branches, and at 2 different places, independent of one another, the bony skeleton developed. Right? So this one is a little complicated as compared to hypothesis 1. Thus, based on principle of parsimony, people believe that hypothesis 1 is worth explaining, right? So, yeah, I have mentioned it over here that the parsimony principle is basic to all sciences, and it basically tells us to choose the simplest scientific explanation that fits the evidence. In terms of tree building, that means that the best hypothesis is the one that requires the fewest evolutionary changes. Right? It might not be true. Maybe evidence comes up that this is actually what is happening. Okay, somebody's trying to join in. Okay, let me just share again. So who had this question? Ajay, was it you? Was I able to address that? I just need to mute this particular individual. Yeah. I will share again. Okay. So I understand when he learns parts of concept, it is actually difficult for us to grab or understand things with a clarity, right? But what I could suggest maybe is, for example, in this particular thing, if you had a doubt, right? So I always mention the references from where these things have been picked up, right? And this Berkeley.edu, like if you have basically gone through Harvard EDX, right? It is one of the courses that is actually offered by Harvard University. Similarly, there are chapters that are present in Berkeley.edu also. One of the chapters was Understanding Evolution, from which I have picked this particular excerpt to basically explain something that I wanted to in conjunction to the question that that had been asked, right? But if you want a clear, complete understanding of this particular thing, what you can do is you can actually refer this this particular citation reference that I have given, and you can go ahead and read thoroughly. Then, okay, this particular question came wherein I was explaining why minute details or understanding very minute statements is basically important, right? So this particular question asks, which one of the following refers to speciation due to geographical isolation? So is it sympatric speciation or parapatric speciation or peripatric speciation or allopatric speciation? So speciation is a concept that was briefly talked about in the course. So owing to that, we will discuss this particular concept today. But if you remember adaptive evolution and your evolution classes, if you have done those, you will basically be able to answer this. So the answer for this particular question was allopatric speciation, right? So what is allopatric speciation? It is basically speciation or evolving of two different kinds of species due to geographical isolation between them. Now, let us go through all these kinds of speciations one by one. So first, let us discuss what speciation is. It is basically how a new kind of plant or animal species is created, right? It occurs when a group within a species separate from other members of its species and develops its own unique characteristics. So if we talk about sympatric speciation, for example, let us first read this and then we'll understand it via what is described over here. So sympatric speciation is actually one of the controversial concepts, right? I have mentioned it over here that it is a little controversial and some people don't believe that it actually happens. So what happens in this particular speciation is that it occurs when there are no physical barriers preventing any members of a species from mating with one another and all members are in close proximity to one another. But a new species, perhaps based on different food sources or characteristics, seem to develop spontaneously, right? So there are no actual physical barriers or no different environmental conditions. No such things are happening, but in an original population itself, there might be some kind of preference changes that might occur due to which there is a spontaneous generation of newer species within the population, right? So that is why over here, when you see there is overlap between the two species that is present, right? So it is a concept that is still under a lot of debate whether such kind of a speciation, that is sympatric speciation, whether it happens or not. But anyway, so this was what sympatric speciation is. Now, coming to parapatric speciation. What happens in parapatric is that the species is spread out over a large geographical area. For example, let us assume a desert, right? Although it is possible for any member of the species to mate with any other member, individuals only mate with those in their own geographical region. For example, we say that we have a 100 acre is very less for any population if we are talking about, but let us assume 100 acre was a lot of land, right? If we say we have 100 acre of a land and in this maybe ants are present in different regions of this 100 acre, right? So although it is not impossible for them to move across this 100 acre, right? But what happens is that they usually they are restricted to niches. Maybe in this 100 acre there is one sugar factory, one sugar cane processing unit, one maybe cotton mill, one is a rice mill, and one is a potato storage unit, right? So maybe there is a set of ants which only are restricted to the potato mill. Just because they are getting food over there, they don't want to go out, right? Or some other who are in the sugar factory. So what happens is that such different habitats influence development of different species in parapatric speciation, right? Instead of being separated by a physical barrier, the species are separated by differences in the same environment. So they are having different kinds of things within the same environment. But because of their preference, they basically get separated. So sometimes a species that is formed by parapatric speciation is specially suited to survive in a different kind of environment than the original species, right? For example, over here they have represented that if there was an original population, there they could have entered a new niche. For example, what happened was that maybe in this 100 acre land, earlier it was completely dry, right? And there were species that were living in this particular land which were okay with it being dry also. But what happened, all of a sudden, maybe a pool was installed, maybe by man, or it formed somehow, like the Grand Canyon formed somehow, right? So maybe a pool was installed. And now some of these species, they preferred staying by the pool, while the others were still okay with just being on the land only, right? So there was no physical barrier that these other species could not go to the pool or these species in the pool could not come out, right? But just because there was a differentiation in the habitat, some preferred the pool environment, some preferred the dry land, right? Due to which there was a distinction in species that was obtained and new species were formed. Then we have the peripatric speciation. So when small groups of individuals break off from larger groups and form a new species, this is basically known as the peripatric speciation. Physical barriers make it impossible for members of the groups to interbreed with one another. In this particular speciation, this one group is much smaller than the other, which is actually the main difference between peripatric and allopatric. Because in both these cases, there are physical barriers that are involved. But if we are particularly talking about a small section or small group of individuals breaking off from a larger group, then it is peripatric speciation. Okay. Unique characteristics of smaller groups are passed on to future generations of the group, making those traits more common among the groups and distinguishing it from others. For example, in case of peripatric speciation, what happens is there was an original population that was present. A small population got separated out. For example, if we say that there was an island and then there was a break that happened between this island and only, for example, if again it was a 100-acre island, 5 acres broke off, right? In this 5 acres, there were only few individuals that were present. But it broke off and now these few individuals have gone and floated off to a different place wherein this 5 acre has formed a new island altogether and this 95 acre is left with the original species and original habitat, right? So when this 5 acres has gone and settled somewhere from where these species are not now able to come back, right? Now these species that are on this 5 acre land will reproduce among one another only, right? And they will maybe develop a different kind of habitat of living. And once people are continuously interbreeding among one another only, there are new kind of changes that will basically be accumulated over here. For example, say that some of these individuals that have broken off were birds. Some of these birds on the mainland, which was a 100-acre land, some of these birds could fly. Some of them had lesser flying abilities and some of them could not fly at all, right? Now the section that has broken off has birds that can only fly on it, right? So once they interbreed, they will only develop offsprings that can also fly nicely, right? So now the other kind of variations that were present within the population has been lost. So new kinds of species will be created. Coming to the allopatric speciation, which was the answer for this particular question, it says that this kind of speciation basically occurs when a species separates into two separate groups which are isolated from one another. A physical barrier such as a mountain range or a waterway makes it impossible for them to breed with one another. Each species develops differently based on the demands of their unique habitat or genetic characteristics of the group that are passed on to the offspring. So, like I talked about the peripatric speciation, allopatric is very similar, except the fact that in this, more or less equal distribution of individuals happened initially, right? So initially the gene pool was almost uniform. There was not such a condition wherein a particular gene pool had basically separated and they had gone and formed a new species just by reproducing, right? That was not the case. There was an actual physical barrier that formed between them. For example, if a Grand Canyon, which was earlier not present, it formed between species, right? Now these species are physically separated, especially the terrestrial species. The aerial ones can still fly from here to there, but the terrestrial species are separated now. So now on these two ends, we might be facing different challenges in life, right? And these challenges might be making them more susceptible to some things and less susceptible to others, right? And they will basically develop into they will develop newer characters, lose some of the previous characters, and they will diverge into new species. Okay. So this is just a summary slide for what we talked about. These are the 4 main kinds of speciations that are known of. The other is artificial speciation, which is something that is basically introduced by man, right? Okay. So coming to question number 8, which one of the following options is true with regard to property of transposable elements in the genome? So is their movement within the genome regulated by the host, or they are absent in the prokaryotic genome, or the amount of transposable elements positively correlates with the genome size of the host species, or all of these? So which one do you think is true with regard to transposable elements?
B.
B, are they absent in prokaryotic genome? Okay, so let us look at the answer for transposable elements. The actual answer is C, which is that the amount of transposable elements basically positively correlates with genome size of the host species. So the first one is definitely untrue, because these are jumping genes and they are not regulated by any regulating machinery, right? The second one, that their absence in prokaryotic genomes, is also not actually true. So there are some primitive kinds of transposons, especially some retrotransposons that are basically found in these prokaryotic genomes, right? Then C is definitely true, because what we say is that in humans, again, I would like to refer back to the concept of the gene density. Like in a 1 kb segment, yeast will have more density of genes than humans, right? So the non-coding sequences, which are not genes over here, could be anything. They could be regulatory sequence, they could be non-coding RNAs, they could be non-coding regions altogether, or they could even be transposable elements, right? And it has been found that this amount basically positively correlates with the genome size of the host. So since we have a larger genome size, but almost equal number of, or almost similar number of protein coding genes, transposable elements is one of the regions that has expanded in us, right? Because we have higher genome size. So transposable elements are basically mobile DNA sequences which are capable of replicating themselves within the genomes independently of the host cell DNA. They also typically range in length from about 100 to 10,000 base pairs, but sometimes are far larger than that also. Briefly, let me introduce some aspects of transposable elements, right? So, broadly speaking, we can divide yeah.
One interesting thing,
Yeah.
I think the concept of transposable genome was hypothesis by Miss Barbara McClintock or something like that lady,
In
which became
cons, yeah.
after 44 years. Means
Yeah, what?
this was a hypothesis by Barbara McClintock. She
Yeah.
proposed this might be the reason for
Yeah.
having this in transcriptonics, but at that time there was no method to prove she was right. So that was a hypothesis.
Yeah. Yeah.
And it became fact after 44 years.
Yeah.
And I think she got Nobel Prize after 44 years.
Yeah.
And same thing with the Mendel. Mendel was just factors were hypothesis. And that becomes fact after 33 years. That's why I was confused with the facts and hypothesis. This is a very interesting. This just come up in the same class. The amount of transposable element and the transposomes were proposed by, I don't know exact name. Sorry, I'm not so much deep in that, but that is a very good.
That is actually true. That is actually true for a lot of technologies, right? For example, there was proposed structures of DNA before the actual structure was actually discovered, right? They were proposed. One of the proposed structure was then also semi-conservative, right? So first it was proposed, and there was no technique available to actually prove it. But later, when these experiments were done, they could actually prove that yes, this proposed hypothesis that DNA is semi-conservative in nature is actually true. So that is the case with most of the things. But when we, with respect to the previous question, that was phylogenetic trees, we are basically saying that we are choosing one hypothesis and we are saying that this is phylogenetic tree, right? Now the point is that the other hypothesis that we rejected, right, while actually making that tree could also have been true. For example, if we again talk about DNA structure, they said it could be replicative, semi-conservative, or conservative, I think. Right? I don't remember the terminology. There were three proposed structures that were given. Right? One out of which was true. But before that was actually scientifically tested, there were papers that were trying to prove the other two structures also. Right? So all these hypotheses were structures of DNA until that hypothesis was proven. Once that hypothesis is proven, maybe it will no longer be the phylogenetic tree, right? Maybe we will name it something different only. So, and there have been some kind of things that have been proven, basically by the presence of connecting links, missing links, that fossils have been found. Carbon dating of these fossils have been done to exactly tell, like, at which age, at what time in evolution these particular things were present. And we know that maybe rabbits, they came much later than that, right? So such kind of things are constantly being done. But the general concept of that is presenting a hypothesis to basically work on, right? Okay. And yeah, very rightly said, Barbara McClinton actually proposed that transposable element, so jumping genes. Yeah. Yeah. Please go ahead.
Screen is not visible.
Oh, it is not visible. I wonder what could have happened. It was visible, right, when I did start discussing this question?
Yes, it was visible previously, but not now.
Okay.
Now visible.
Oh, all right. Yeah. So, yes, this concept was actually brought forward by Barbara McClinton, who was, yeah.
Sorry, madam, but
it's not visible. Okay. I really don't know what the problem is over here. I also checked on my other device. It is actually not visible. Okay. Let me just try and join again. Once I will try to share again. If it is not working, maybe I'll leave the meeting and I'll join again. So for now, we know it is visible, right?
visible. Okay.
Yeah. Okay. Yeah. So corn kernels were what she was actually working on, and this was initially proposed in corn kernels because different kernels had different colors. So that is also actually a very interesting study. Um, Maybe you all can go and read more about it also. If we broadly speak about transposons, they can be divided into two classes, right? So first, let us discover what is over here on the left-hand side, right? The first class of transposons is known as the retrotransposons. So anything that has retro in it, what does it mean? That it basically forms DNA from an RNA, right? So reverse transcription, retrotransposon, all these things, RNA intermediate is required, and RNA basically leads to formation of DNA. So what this retrotransposon class of transposons actually did was that these were present between our normal DNA. So this blue segment represents our normal DNA, and the this orange color in between represents a retrotransposon or class 1 transposons. What happens is that they direct the transcription of an RNA, right? So from here, RNA polymerase, it basically comes and binds and it directs the transcription of an RNA. Once this RNA is made, what happens is that this RNA through reverse transcriptation, transcriptase undergoes reverse transcription, which basically forms it into a DNA. Now, this new DNA that is created goes and it basically sits on any other target DNA, right? So maybe this is another target DNA between which it goes and sits. So it is a transposable element. While in the second class of transposons, that is known as DNA transposon, what happens is that For example, there are these two segments of DNA that are present, and this green colored region is the transposon. This transposon gets excised from here, right? So it gets detached from this particular DNA regions, and this particular DNA goes to and binds to another target DNA. Right, so it integrates between another target DNA, and this also basically represents transposable element. So over here in class 1 transposon, what is happening? It is basically referred to as a copy and paste mechanism, right? I forgot to mention it over here. It is one of the interesting things. This is copy and paste mechanism. Why? Because this original transposon will remain over here. It will be copied into RNA. RNA will again form DNA, and this new DNA will go and sit on another target DNA, right? So at two places now it is present. It formed copies, right? While in this particular DNA transposon, what happens? It is cut and paste mechanism, right? So from the first place, it was cut, right? It was cut from here. Then this excised DNA basically went and it sat on this other transposable element. So now it just has one copy. First it was sitting over here, and now it is sitting over here, right? So these are two kinds of mechanisms if we broadly speak about transposons. So these two things happen. Now there is a very huge subclassification of these retrotransposons and DNA transposons, into which we won't be getting in this particular lecture. But yeah. Okay, the screen is not visible. Just give me a moment. Maybe I'll join again. Maybe that resolves this, right? Yeah. It is still recording. Let me try and share my screen. No, it is not sharing, right?
Visible for Jeffrey, gone.
No. It was not visible. I'll try again.
Now visible.
Now I suppose it is. Yeah. Okay. Yeah. So this was basically the difference between retrotransposons and DNA transposons. Okay. Also, what I was mentioning was that they are further divided into different kinds of subclasses, line signs, ALU elements, P elements, right? Into which we won't be getting right now because that is a huge topic of discussion altogether. But over here I have. Yeah.
One in this
Yeah.
cut and copy and paste. First, copy and paste. So it is not a complete copy and paste. It is a rewriting because uracil and thymine will be different in RNA, and it will be the RNA dependent. Means RNA dependent polymerase will do, and here it will be DNA dependent DNA polymerase. But in that DNA, yet both strands are added, or one of the strands of DNA is added. Means just I want to know कि RNA intermediate and DNA intermediate only have that methylated difference of thymine and uracil, or because it is both of the strands of DNA intermediate are added in target DNA. That's why it's a different from first one.
No, actually, as far as my knowledge about this goes, it is not compulsory that a particular transposon is present in two copies, right? So even in DNA, it might just be present in one copy, and that one copy will go and it will basically sit between another target DNA. So in both these, one copy is only playing a role, and in this particular case, that is a DNA transposon, the DNA polymerase actually has no role to play. because it is not
Then-
being copied. Yeah.
Suppose there is one, then it cannot be cut and paste. It will be also copy and paste. It have to be both strand. Otherwise, it will again regenerate in the original strand also. From the
No, so
remaining.
there could be looping out of the strand and removal of it altogether.
That's what I think. Maybe means
Yeah.
my hypothesis. Maybe your hypothesis.
Yeah. So, in some cases, when a region is generally deleted, what might be possible is that, in one case, what is possible is that the DNA repair mechanisms comes and takes over, right? In other cases, what is possible is that, for example, this was a DNA strand and its complementary DNA strand was present, right? This region was lost. The one that I have not made over here, this region was lost altogether. So another mechanism that could happen was that when this particular one,
Can I try to make it simple? If a part of DNA means means one of the two strands, means if part of one strand of DNA is there, then the first mechanism will obey. If the part of both DNA from the same site is cut, means AG, TG sites is cut, then there is second mechanism will follow.
okay.
But there is a only means single stranded part is there. Always there is a use of reverse transcriptase and the whole mechanism follow in that way. And that's why it is written in the original DNA also. And when both the sites they are cut, it cannot be written in the original DNA. So what I think is in first part only the part of single strand of a DNA is cut. In second, the part of both strands of DNA are cut.
Okay. I actually will have to go and check this particular part, but the machinery for actually introducing it at another place is also different. There are two major differences that still happen over here. One is basically that the original copy in the first class is actually retained at the site, while the original copy in class 2 transposons is lost at the site it was originating from, right? Second is that there is the use of both RNA polymerase and reverse transcriptase in the first case, whereas there is no utilization of any polymerase or transcriptase in case of the second one. But as far as this particular thing is the focus, that whether or not the other strand also has the same thing that is going on over here, that is something I will have to check. Okay, so that I'll check and I'll get back. Also, with respect to this transposable elements that over here have been mentioned as TEs, there was this interesting paper that I got, which I suggest that you guys read. So this is a very brief outcome that the paper gives. It is one of the schema that I have just pasted from the paper, which basically defines the different kinds of basic basic information and how that we basically know about transposable elements, right? So that they are present in different forms and shapes. They are not really randomly distributed. So there are some places wherein there is a tandem repeat of or continuous repeat of transposable elements. On other places, they are not at all present, right? Then they might be sources of mutations or genome rearrangements. They might lead to expression of a gene or a repression of a gene wherein they are getting introduced. They affect both germline and somatic cells. Also, they might induce immune responses because they are capable of making proteins also, non-coding RNAs or proteins. They are capable of making them, right? Also, they create genes and RNAs. Then they can modify regulatory networks by basically setting on a specific regulatory sequence. And they also require special tools to jump from one place to another, right? So this paper summarizes very beautifully. It is not a very long paper. It summarizes very beautifully how the knowledge about transposable elements that we currently have. I really urge you to go and read this paper. It will help better your understanding about this also. And the mechanism part, it does not really talk about that. I will check from one of these traditional books that we have, and I will let you know. Okay, so coming on to question number 9, among the options given below, identify the group of databases that cannot be used to analyze human genome sequence. So it is a very simple question because of the options. The first one has options: ensemble, encode, UCSC genome browser. B is encode, gene atlas, and ensemble. C is GeneCard, RefSec, and Gene Atlas, and D is encode, Gene Atlas, and Flybase. So the answer to this is very simple, because Flybase is basically a database for fly or Drosophila melanogaster genome, right? So that will not be helpful for human genome sequence. So thus the option is D over here. Coming on to our final question, which is question number 10. It says phylogenetic trees are often constructed by comparing genome sequences of diverse species to find their relatedness. Based on your understanding, state whether the following statements are true or false. So the statement 1 says a large number of differences correspond to less relatedness among species, while statement number 2 says smaller number of differences correspond to more relatedness among species. So which one do you think out of both these are true? Are both of them true? Are both of them false? Is statement 1 true but statement 2 false? Or is statement D? Yeah. Which one?
D.
No, so that is not actually true. Over here, we can say that larger number of differences will correspond to less relatedness among species, because that is how we are actually building our hypothesis, right? So we are saying that if there is lesser sequence similarity, they will be less related. If there is more sequence similarity, they will be more related. Right, that is what we are saying when we are basically constructing phylogenetic trees. So, based on, yeah,
or something like that.
what?
These are principles of Boveri or something like
that? I did not understand what you were saying.
These are the part of 1, 2, 3 principles of scientist Boveri. It was given by Boveri.
Oh, okay. With respect to phylogenetic trees, it was not given, right? Boveri was present before we could construct, I think, phylogenetic trees. I don't really remember since when we've been constructing those. At least using MSA, it came much later on, right? Over here, what the question asks is that whether phylogenetic trees are constructing using these principles or not, right? Whether it is true when we are constructing phylogenetic trees. So in general, if we talk, it may or may not be true. Both the statements may or may not be true, right? But when we are talking in with respect to phylogenetic trees, that is what I explained, that is how we are hypothesizing. How we are hypothesizing that maybe apes and humans are close by, and maybe dinosaurs and humans are not so close by, and apes and fish are again present at different distances from each other. How we are basically hypothesizing that is based on the sequence similarity, right? So in phylogenetic trees, when we construct them, we believe during construction, we hypothesize that larger number of differences correspond to less relatedness and smaller number of differences correspond to more relatedness, right? So that is how we say that humans and chimpanzees are very similar, because we share about 99% of our DNA, right? You and I are more similar because we share about 99.99% of our DNA, right, with respect to sequence similarity. So that is how phylogenetic trees are hypothesized and constructed, right? So that
So,
is it
madam,
for the
that's
first. Yeah.
parsimony, like the same thing you had explained previously. So same, it is means principle of parsimony. Ki simplicity is there, or in the hypothesis you had explained. That's what I am asking.
Yeah. Over here also, I said the same thing, that we are hypothesizing when we are building phylogenetic trees, we are hypothesizing that larger number of differences correspond to less relatedness and smaller number of differences correspond to more relatedness. That is also a hypothesis, right? I am not saying that this is the principle over here. I am saying that while constructing this, this is our hypothesis. Because based on that, we are building the phylogenetic trees.
Okay, madam. Thanks.
Yeah. Okay. So moving on to the second part, which is new advancements in genome editing. Over here, like I mentioned, I have briefly talked about base editing, prime editing, and paste, which is another newer form of introducing or inserting larger number of base pairs into DNA without double-stranded breaks. So before this, before beginning with this presentation, there is a disclaimer that I myself, I am not very well read on these particular techniques, right? So for this particular class, I have read a little about it and I have compiled it as a presentation, but I am not well read about this. So I urge you I will be able to explain the principle that goes behind it and how it might be better or worse than some other techniques. But any technical details I might not be very well versed with. So I urge you to go back and look at these discoveries, which are relatively newer as compared to CRISPR maybe or other genome editing technologies that we talk about. And yeah, so that was the disclaimer from my end. So I hope it is still visible. I'll be beginning with this presentation. So when we talk about genome editing, one of the major techniques that we did discuss about was CRISPR, which I'm talking about within the newer techniques. So we did talk about Kree-lock systems, inducible systems, conditional knockouts, then just knockouts, right? These things also we discussed about. But in the newer systems, what we talked about, the latest one that we actually discussed in this course was CRISPR, right? So CRISPR has actually revolutionized the field, and it has revolutionized the field to such an extent that there are a lot of techniques that have been built upon this basic technique, right, which is CRISPR. So let us look at these particular techniques. First, before we actually move on to them, let us understand how the double-stranded break, which was being introduced by the Cas9, right? When Cas9 recognized it, basically introduced a double-stranded break. That is why it was known as molecular scissors. So it was very convenient for research purposes. But why was it a concern as well as convenience, right? So first, let us generally talk about how double-stranded breaks actually helped us. So genome editing modalities involve generation of a double-stranded break or DSB at the target site. Once the double-stranded break occurs, the researchers take advantage of cellular DNA repair mechanisms to abrogate gene expression by a gene knockout or to introduce specific modifications such as correcting a point mutation back to its healthy form. So this was what was being done when researchers were using HDR or NHEJ mechanisms to actually direct mutations within the kind of mutations that they liked in their models, right? So this was what was happening over there. Over here, we say that double-stranded break, although it helped achieve these objectives, it was also a little troublesome. Why? Because genetic lesions could basically affect the cell, right? And if it was not properly repaired, it could have extreme consequences. There were concerns linking double-stranded break response and CRISPR that have risen in particular after two publications that were basically published in 2018, which highlighted how CRISPR editing may select for cells with aberrant p53 expression. So if you are not aware about p53, it is basically an oncogene. Oncogene means a cancer-causing gene. So there were two famous publications in 2018, which basically highlighted how basically if there were CRISPR editing that was being done, if it was not very specific to the site, if it had off-target effects, it basically led to selection of certain cells with incorrect p53 expression. And if there is an aberrant p53 expression, these cells will be very, very susceptible to cancer, right? So thus, after this, it was kind of clear that a double-stranded break-free approach was highly desirable to make CRISPR technology further safer, right? So in response to this, what researchers did was that they basically started to develop newer technologies wherein this double-stranded break did not have to be made in the first place, right? So in the first technique that I mentioned over here, which is the base editing, what happened was that base editing basically combines the powerful DNA scanning and sequence identification capabilities that were present in the CRISPR-Cas9 system with a deaminase enzyme, right? So we will discuss about what this deaminase enzyme will do, which basically introduces a single nucleotide polymorphism by chemically altering the target DNA sequence without intentional generation of a double-stranded break, right? Over here, as the name suggests, it is base editing, right? So a single nucleotide base will be edited. How it is done is that the base editors generally contain these two separate proteins, right? One was CRISPR-Cas9 related system, and the other was this deaminase enzyme that we talked about. So these over here are known as Cas9 nickase. Why is it known as Cas9 nickase? Because Cas9 over here has been modified through mutations in one of the two amino acids, which is responsible for DNA cleavage activity, right? So now this nickase, N-Cas9 or Cas9 nickase, is able to pair with a guide RNA. It is still able to pair with a guide RNA and it can go and find a complementary DNA sequence, right? But instead of creating a double-stranded break, it can now only make one strand of DNA, right? Also, this nucleotide deaminase that we talked about, what it does is that deaminase is an enzyme which is capable of removing amino group from a specific type of nucleoside, right? So deaminase fused to nickase, that is this Cas9 nickase, will dictate whether the base editor is a cytosine base editor or an adenine base editor. Because if cytosine has this particular amino group removed from it, it will convert into a tyrosine. If I the tyrosine, I'm saying adenine, guanine, cytosine, thymine, right? And this A will basically convert into a G. So these basically contain cytosine deaminases, whereas ABs typically contain adenine deaminases. How it works is basically that as we talk, there is still this Cas9 machinery that is present. This Cas9 machinery, with the help of its guide RNA, goes and recognizes the complementary strand, right? When it goes and recognizes this complementary strand, it can nick only one part of the strand and it will not cause a nick to the other strand, right? Now, this other strand will instead have a editing window because it is open, right? So, because it is open, there will be a 5 to 10 base pair editing window that will be present, right? So, okay, C will change into T and A maybe will change into G. Okay. So in this editing window, what will happen is that among these 5 to 10 base pairs, if it is a CPE, cytosine base editing deaminase, then it will edit the base cytosine to thymine over here, right? On the contrary, what will happen if there is an ABA or adenosine base editing, then it will basically convert adenine to guanine by deamination, right? So this is how this deamination works. And what happens after this is that once this particular site is edited, once the complementary DNA comes back, the other base also through repair mechanisms, it is basically changed to a complementary site to either thiamine or guanine, right? So now there is change from C to T in one strand, and on the complementary strand, there will be a change from G to A because of the DNA repair mechanisms. That is how it will work. So from this base editing, what was achieved was that now double-stranded breaks were not made, right? If the double-stranded breaks are not made, the the kind of aberrant cell expression of oncogenes that were being seen is solved to some extent, right? Why I'm saying some extent is because still one strand, the nick is actually happening. In the other strand, although it is not happening, in the first strand, the nick is still happening. So, although it was an improvement over the previous technology, which was CRISPR-Cas, Cas9, it still had some limitations. Firstly, it required precise positioning of the base editor edit window, right? The edit window had to be very precise because that edit window only was for 5 to 10 base pairs. Now, in that edit window, if there are multiple cytosines that are present, and we just want to edit one cytosine, there will still be unwanted edits in other cytosines that will happen, right? Because we are giving a cytosine binding base editing deaminase, right? So there will be other cytosines also that might be able to be edited. So either you have to design it such that you have minimal number of cytosines that are present in that target region, something like that, right? Then the generation of insertion deletion was not possible in this. So if our aim was to actually insert or delete some sequence of DNA or DNA, then that we will not be able to do using base editing. Why? Because over here double-stranded break is not happening, right? Also, there was some off-target RNA editing that was happening when we were using this base editing technique. And this base editing had come out in around 2016. Yeah, so this is the actual paper in which they had proposed this base editing. It had come out in 2016. After this particular group only improved upon the technique and they basically brought in prime editing. So it was by the same group, although the first author is different because the PhD student might have been different, but the lab was the same. So it was Professor Liu's lab. I'm missing the institute, but you can refer this paper for that. And what they did was that they basically brought in a new editing technique. Now, this editing technique was known as prime editing. What prime editing did was that it is again a CRISPR tool, right, which allows broad manipulation of genomic DNA without requirement for potentially deleterious double-stranded breaks. So over here also double-stranded breaks were not happening, right? Instead, over here, again, the Cas9 nickase was actually combined with a reverse transcriptase. So what this basically did was that it made special use of a guide RNA named PEG RNA. So what this PEG RNA did was that it acted both as a GPS that directed the Cas9 nickase toward the genomic target and as the template itself. And it also acted as the templated cell. So I think that is a missing thing over there. Then, beside possessing the sequence complementarity to its target, the PEG RNA also included a RNA donor sequence. So what this donor sequence did was that once the Cas9 nickase cut the single strand of DNA, the donor component can be used as a template by the reverse transcriptase fused to Cas9 nickase, which will transcribe a complementary DNA strand based on the RNA template included in the PEG RNA. This newly synthesized cDNA will contain the desired modification and will be installed where the nick occurred. By programming the design of the PEG RNA, it was possible to introduce insertions, deletions, and even small changes in the DNA sequence. So what was the basic principle of this? In this, they have basically used a special reverse transcriptase, right? What the special reverse transcriptase actually did was that it guided the Cas9, it created a nick, and after creating a nick, it also had a template, right? This template had whatever desired deletions or insertions or kind of sequence that we wanted to be inserted into the genome, right? Now this after nicking the strand, it basically presented this template to that nicked strand, right? And according to this template, a new strand was formed where this nick had occurred, right? So now this new strand that has formed will basically contain whatever we wanted in that particular genome, right? So over here, okay, this should have been my first slide. So over here, what happened was that the prime editor complex included Cas9 enzyme. This Cas9 modified to only nick one strand. I should zoom this. Modified to only nick one strand of DNA and a reverse transcriptase enzyme, which can generate new DNA by copying an RNA template, right? Then this engineered PEG RNA, which is prime editing guide RNA, sends the editor to its target where Cas9 nicks the DNA. After Cas9 has actually nicked the DNA, what will happen is that the PEG RNA again has two special components, a section that will bind to the nicked DNA, preparing the nicked strand to have a new DNA letters added, and another section of RNA letters that encode the desired edit that we want. This is a desired sequence that we will want, right? It is represented over here as binding region and edited sequence. Now, to transfer the edited sequence from PEG RNA to the target DNA, the reverse transcriptase reads RNA and attaches the corresponding DNA letters to the end of the nicked DNA, right? After this would have been added, what will happen is, okay, let me zoom in. An endonuclease in the cell naturally would excise the old segment of the DNA and seal the new letters into the genome. Now, once this old segment has been excised, now what do we have? We have a mismatch, right? Because the target site is left only with one strand and one unedited strand, right? One is an edited strand and the other is an unedited strand. So, to resolve this, the mismatch favoring the permanent installation of the edited DNA, a different guide RNA directs the prime editor. Okay, so it is not just the repair mechanism. I misspoke this, that there will be another guide RNA that will basically direct Cas9 to just cause another nick on the other strand, right? This will be caused by this other guide RNA, but it will not have another sequence, right? So over here, like we said, it had an edited sequence. That edited sequence will not be present over here. It will just have this nicking property, and this nick will basically prompt the cell to remake the nicked strand using edited strand as the template. So this first strand will be used as template, as thus completing the edit. So now we would have a fully edited DNA. So this is how the prime editing would work. And as I spoke, this also came from the same group that actually made the base editing tool, right? So from that, you can basically link that over here also. Although there was not a double-stranded break that was happening, there was still a single-stranded break that was still occurring, right? So it will still pose some difficulties and some off-target effects, right? In order to further better this technique, we have a very new technique which is fresh from this year, that is 2023, from another group, wherein they have basically made another kind of tool. Why did they want to make this tool? Was that programmable genome integration of large diverse DNA cargo without DNA repair of exposed DNA double-stranded breaks was an unsolved challenge in genome editing before this tool actually came in, right? So for this, what they have done is that they have basically made a tool that is known as PASTE. What does PASTE stand for? It is a programmable addition via site-specific targeting elements, right? What it does is that it uses a CRISPR-Cas9 nickase, like which was basically made by these individuals in the previous papers that who had discovered base editing and prime editing. So over here, they had used that same Cas9 nickase, and they had fused both a reverse transcriptase and a serine integrase for targeted genomic recruitment and integration of desired payloads. So over here, what is the target of these people was to just paste a segment of DNA. So they said that we will not cause any breaks at all, whether a single-stranded break, double-stranded break, no breaks at all, right? We will just take whatever has to be introduced and introduce that particular thing over at the desired target site, right? So this actually I have taken from their paper only because there were not many representations that have come out as yet because it is a relatively new technique. So what they had done was that they had basically taken again a Cas9 nickase. So this protein that you can see is a Cas9 nickase. It has been fused with an integrase. Now this integrase is named as BXB1 integrase, right? Along with this, there are other things that they have taken. One is a human genome target, right? So this target is within a human cell maybe, right? Apart from this, there is a nicking guide that is present and another guide that is present which has a guide RNA and an ATT-B site. Now this ATT-B site is an attachment B site, right? So what they have done is that first in this particular human genome target through Cas9, they have basically introduced the addition of integration site. Right, so integration site, that is ATT-B site, has been introduced over here. That is this particular tool. So this they have just done by a normal CRISPR mechanism. For example, a prime editing tool only they have used, and they have introduced a small ATT-B site in this particular human genome target. Now, once this site was introduced, what they have done is that they have taken this particular protein which contained the gene and the ATT-P site. So this attachment P site basically goes and attaches to the attachment B site, right? And along with it, it has the gene, right? And both these are carried on the integrase, which is basically integrated upon a Cas9. So what will happen is that this Cas9 will basically go and recognize whatever target DNA it has to recognize. After recognizing this target DNA, what will happen over here with the help of integrase? Over here, there are no nickase that is present, right? Or no molecular scissors that is playing any role. Over here, there is an integrase. This integrase will go and it will basically find its target, which is ATT-B, right? So this ATT-B will be found by this ATT-P site that is present on this integrase, and once it finds this site, it will attach to this ATT-B such that this gene is basically integrated within that particular genome target, right? So this is basically a pasting mechanism, which is one of the newer editing tools that has been devised by this particular group. Now, what is the advantage of this pasting mechanism? So sometimes what happens is that maybe a gene is not functioning because its regulatory sequence is missing or defective, right? So over there, only if that regulatory sequence is added, right, it could just work normally like it would in any other individual, or similar case for a gene also, right? Maybe a gene does not have a particular exon because of which it is not working. We could introduce that exon and it will have a normal function. Or even for creating transgenic animals, it could be very useful, this technique, right? And what they said was that in case of PEG editing, PEG RNA, they could still introduce some edits, but it could only be for a maximum of 4 kb. 4 kb. 4 kb. But over here, they said that they could introduce segments ranging from 3,600 base pairs to 36,000 base pairs, that is 36 KB, to up to 50 KB. It has the potential for up to 50 KB. And what this group has also done is that they have themselves gone and checked the metagenomic databases, from where they have found a number of integrases, combinations of which could be used to target different kinds of cells and DNAs in various individuals, right? So that is also something that they talk about in the paper. So this was a general outlay of the kind of thing that they have designed. So that is basically the end for these 3 editing tools also that I had to introduce. I would also like to give you an update of another tool, which is not a genome editing tool per se, but it is something that is being used for diagnostics now, right? It's not yet being used, but it has been proposed to have a lot of diagnostic potential. So that particular tool, like that particular paper came out in August itself, I think on 10th August, and the name of that particular tool is Catch, CATCH, Catch. So in this tool, what they do is that they have basically engineered through use of horizontal gene transfer and CRISPR-Cas9, they have introduced a bacteria, right? So what this bacteria basically does is that now, because of CRISPR-Cas9 system being a part of it, it can recognize whatever target it has to recognize, right? And because of the horizontal gene transfer, what they have they have done is that they have introduced certain spacer sequences within this particular engineered bacteria, such that it can go and it can identify certain cancer mutations, right? So for example, they have said that they can use this particular engineered bacteria to identify mutations in cancers, right? So for example, there was a patient, and this particular patient has colon cancer. Right. So for colon cancer, they would still have to, for most of the cancers, people have to undergo biopsy, right? And biopsy is a very tedious process, and sometimes the cancer might be at such regions wherein biopsy is not possible, right? Or the cancer has spread such that there, like, where all will you do biopsy in the first place, right? So in such cases, there is cell-free DNA that was being tested in these individuals, right? What the cell-free DNA was doing was that in the blood of the individuals only, like sometimes cancer cells leave some markers that could be present in the blood or stool or any other such place of this individual, right? It is not restricted to the cancer cell, through which they were trying to identify whether this person has cancer or not. But that technique has very less reproducibility, or those tests are very, very costly because they are PCR based. While what they have done over here is that they have basically engineered this bacteria which can recognize a particular mutation, right? For example, if we know that colon cancer has these 5 major kinds of mutations that are present, so we have basically devised a cocktail of such microbes which can identify various such mutations, and we have introduced it into an individual. So because the study has not been done, I do not really know how this introduction will be possible, right? But what they propose is that once it is introduced into an individual, what it will do is that it will go and detect only the mutated cells, right? If it can go and detect only the mutated cells, when we take the stool sample of this individual, if we are talking about a colon cancer, this stool sample can be cultured on agar plates, right? Agar plates. And overnight culturing is required. The next day, these bacteria, if they have actually identified the mutation, they can show that GFP positive signal, IRFP positive, any such positive signal that will tell you that, okay, this particular mutation is actually present in this particular individual. So this person has this colon cancer because of XYZ mutation, right? So such things are possible, and if it will be possible, and if it will be brought into clinical trials, its reproducibility is very high because it does not require cumbersome techniques like PCR, qPCR sequencing, right? You just have to culture that particular sample and you can find out. The only thing that you require is an engineered bacteria. So the process is very simple after that bacteria has actually been engineered. So that basically brings us to an end of this particular lecture. And thank you for being here. Also, that brings us to the end of this particular course as well, right?
Thank you.
Yeah.
Very much.
Yeah. So, if you guys are interested, I really urge you to go and read upon such genome editing techniques. Obviously, there are other techniques also. These were the major ones that have come up. There are other such techniques that keep coming from time to time, right? So, it might really help you or interest you in this field. So, with that, I would like to take a leave and happy learning to all of you.